The reason that I became really interested in cheese is because I was hoping to find a system where I could study microbial communities. As we know, microbial communities are everywhere and they're incredibly important for pretty much everything on the planet, whether it is the ocean or soil or the human body. But the challenge with understanding how those communities work is that they're so complex.
Working on cheese provides this incredible opportunity to move from looking at patterns of diversity, which we can do in lots of microbial systems, to actually begin to test where those patterns come from.
What we did in this paper was to try and take this in situ to in vitro approach. So we started looking at the microbial ecology of these communities. Then we brought these communities into the lab. We were able to culture all of the dominant members of these communities. And then with that culture collection, we were able to reconstruct communities in an in vitro system. So basically, with the system, we can go from an in situ community all the way through to an in vitro system to understand how these communities form and what the interactions are between the species.
Most other systems, it's incredibly difficult to culture all of the microbes that are found in that system, bring them into the lab, and then recreate those communities. And we're able to show that we can do that.
Surprisingly, even though cheese is something that's been made for thousands of years, we still don't really have a really deep understanding of what species of microbes are actually found on cheeses and how much they differ from cheese to cheese. We are studying the rind of the cheese, which is the community that forms during the aging process. So when you take milk, you make it into a fresh cheese, and then you can place that fresh cheese into some sort of aging environment, whether it's a cave or a temperature and humidity controlled room. And during that aging, you get the formation of this microbial community or biofilm on the surface of the cheese. And so what we did was we collected these cheese samples and we scraped off the rind of the cheese, which is this biofilm that we're interested in. And we sequenced using Illumina sequencing both the bacterial ribosomal RNA and the fungal ITS or internal transcript spacer to look at the taxonomy of the species that were present in each of these communities.
To me, one of the most impressive things about the system is the idea that you can have a cheese made in France, a cheese made in Vermont,
and for example in Wisconsin,
and a cheese made in California. And if they use about the same kinds of conditions, you'll have about the same types of microbial communities, which suggests that if you build a particular environment, you can select for a particular type of microbial community, which also suggests that there are some underlying features of these communities that are consistent over huge geographic regions.
So we set out to try and culture all the dominant species from these cheeses. So we looked at the organisms that were greater than 1% abundance in our samples, and it turns out we were actually able to culture representatives of every single one of these genera. So we have now a collection with isolates from 10 different fungal genera and 14 different bacterial genera. The fact that we have both fungi and bacteria makes it a sort of interesting system to look for interactions that are involved in community formation.
We were looking for indications of bacterial fungal interactions, both positive and negative, and we found a lot of both. And what was really interesting to me is that it seems like the fungi in the system are driving the growth of the bacteria. So when we grow the fungi and bacteria together, we see the bacteria highly responsive to the fungi, but the fungi don't really seem to care that much about the bacteria, which suggests that the strong players in the system are probably the fungi.
So what we do is we make in the lab what we call in vitro cheese. So this is just freeze-dried cheese curds that we pulverize and make into an agar media. We can then pour this media into plates, and then we can take from this culture collection we have and mix together any combination of organisms we want. One of the other things that we've done in this work is to follow community formation in an actual cheese and then recreate that in the lab. So we went and worked with a cheesemaker in Vermont where we collected samples of actual cheeses aging in caves and sequenced them to see this beautiful succession of bacteria and fungi. And taking our culture collection in the lab, we're actually able to mix these organisms together, and we were able to follow the formation of these communities.
I think many of the things that we see in terms of both patterns of diversity and also thinking about what mechanisms structure that diversity are very clearly applicable to other types of microbial communities. So, for example, we see very clear patterns of succession over time on these cheese rinds forming, and we know that succession is a trait that's found in many microbial communities. And what we're able to do with this system is begin to dissect what drives that pattern of succession.
We hope that this will really be, in many ways, a model for other communities. The idea is to be able to learn some of the basic principles of microbial community formation in this very experimentally tractable system.